Hi, everyone. My name's Sally Potter. I'm going to be moderating today's webinar. And I just want to begin today by acknowledging the Darkinjung people, the traditional custodians of the land on which I meet with you today, and pay my respects to their elders past and present. And I extend that respect to Aboriginal and Torres Strait Islander peoples here today. It's my absolute honour to introduce Professor Jun Kitano, who runs the Ecological Genetics Lab in the Department of Genomics and Evolutionary Biology at the National Institute of Genetics in Japan. Professor Kitano, he did his Ph.D. in Physiology at Kyoto University in the School of Medicine. And then he had multiple postdoctoral research fellowships, both in Japan and the USA. He was an assistant professor in the Department of Ecology and Evolutionary Biology at Tokohu University in Japan. And that was before joining the National Institute of Genetics in 2011. His research takes an integrative approach across diverse disciplines, combining both experimental and empirical research of natural populations to explore the mechanisms driving phenotypic variation, adaptation, and speciation, topics I'm really excited about hearing about today. His lab combines both in vitro and in vivo research to investigate the molecular and physiological functions and to understand the mechanisms that underlie the evolution of biodiversity. He works on the model system of stickleback fishes, which he's going to talk to us about today. And his research has really advanced our knowledge around the molecular mechanisms driving adaptation and speciation. So it's my pleasure. I'm super excited to introduce Professor Kitano today and hear about your research. So thanks for taking the time to present to us.
Thank you, Sally, for a kind introduction. And thank you for inviting me to this seminar series. So I'm Jun Kitano from the National Institute of Genetics, and which is located about 1 hour from Tokyo. So if you have a chance to visit Japan, please let me know. And since 2011, I have my own laboratory, and we are interested in identifying causative genes and the mutations for adaptation and speciation in nature. So in nature, we can observe biodiversity, and we are interested in how such biodiversity has evolved in nature. So I think that the information of causative genes and mutations can help to answer all the questions in evolutionary biology. For example, do evolution occur in few large steps or many small steps? Or when we observe convergent evolution, do different lineages follow the same pathway or different paths? So to this end, we are trying to link causative mutations occurring in natural populations and fitness in natural environments. So we established this research strategy. So anyway, we conduct a field study, so collect mainly fish, but collect fishes from various environments. And we characterize their phenotypes in the laboratory. For example, we measure morphology or measure hormones or the physiological traits, and we sometimes observe their behaviors. And sometimes we cross different populations or species. And once we identify some interesting phenotypic differences or isolating barriers, next step is to conduct some genetic and genomic analysis. For example, QTL mapping, or by using genome sequencing, we can do GWAS, genome-wide association study, where we can conduct transcriptome analysis or chromatin analysis, such as ataxic or cut and tag sequencing. And once we identify some interesting candidate genes, we study their functions in vitro, or we can manipulate genes in sticklebacks or medaka fish, so we manipulate their genes. And the ultimate goal is to conduct ecological experiments to see what kind of ecological conditions promote the spread of certain alleles. So basically, evolution happens by mutation, and that mutation, when that mutation spread in a population, that phenotypes can be fixed. So we want to connect mutations and fitness in nature. So we are mainly using a stickleback fish as a model system. And this fish is a cold water fish. So they are basically living in coastal regions of cold environments in the northern hemisphere. And unfortunately, there are no sticklebacks in Australia. And importantly, in these regions, especially in North America and Europe, these regions were covered by ice during the last ice age. So it is considered that most of the stickleback fish were living only in marine environments. But once the ice was gone, about 10,000 years ago, many freshwater environments became available and they colonized these regions. And they diversified to adapt to diverse environments. And Japan was not covered by ice during the last ice age. So we think that a different scenario of freshwater colonization occurred in Japan. So this is a cold water fish. So during the interglacial period, most of the sticklebacks were living only in northern areas. But during the ice age, they can go to the southern regions. And in some cases, they colonize freshwater environments. And when, again, interglacial period comes, they cannot live in southern areas, but there are some spring water fed cold environments in freshwater. So in such regions, they can survive. So by repeating this process, multiple waves of freshwater colonization happened in Japanese archipelago. So again, in Japan, we can observe phenotypic diversification, not only in morphology, but also in physiology, as possibly as adaptation to divergent freshwater environments. So we have many replicates of such phenotypic diversification. So this is a great system for studying the genetic basis for phenotypic variation. And recently, we have also developed several genome editing technologies. We can, of course, knock out genes, but also, for example, we can induce inversion or large deletions at least 1 megabase by CRISPR. And we are also setting up some ecological experiments. So, for example, in our institute, we established this mesocosm. So we manipulate some environments to see what kind of genotypes are favorable and have higher fitness in certain environments. Or we can set an enclosure in natural environments. And we hope that this kind of experiment can tell us what kind of genotypes have higher fitness in certain environments. So in this way, we can connect causative mutations occurring in genome and ecological fitness in natural environments. So by using this system, we are trying to answer several questions, but today I just focus on one specific question. So today we'd like to discuss how predictable or contingent is the genetic basis for convergent evolution. So I'll show you two case studies that we conducted in my laboratory. So first one is freshwater adaptation. So, you know, fish, as for fish, there are marine fish and also freshwater fish. And a recent phylogenetic analysis suggests that ancestral state is marine fish. But in multiple lineages, freshwater colonization happened, and some becomes freshwater fish. But interestingly, this happened multiple times, but this happened only in a limited lineages. So we think that there should be some genetic constraint, and some genetic factors are necessary for making marine fish to freshwater fish. So we are interested in the genetic basis for that freshwater adaptation in fish. So we think that Japanese stickleback is a great system for answering this question. So first, we collected stickleback belonging to genus Gasterosteus from different regions in Japan, and we made a phylogenetic tree by using a whole genome-wide SNP. So you can see there are two species. One is Gasterosteus nipponicus, which we call Japan sea stickleback, which is endemic to East Asia. And another species is Gasterosteus acreatus, which is a three-spine stickleback. And this is very prevalent, common in Northern Hemisphere. So this is distributed not only in Japan, but also in Alaska or North America, Canada, or Russia, or Europe. And these are different species because we have data that they differ in morphology, and they differ in mating behaviors, and they also differ in karyotype. And once we cross them by artificial insemination, and one direction of hybridization results in hybrid male sterility. So they are different species, but still we can cross them in different direction, and hybrid females are fertile, they are fine. And our genomic analysis suggests that they diverged about 0.68 million years ago, but they are still exchanging some gene. So they're still maintaining gene flow. And this is not directly related to freshwater colonization, but recently we found evidence that strongly suggesting that they are different species. So in 2011, you may know that there was a big earthquake in Tokohu area, and it was a very sad news. But we thought that how can we make this sad news into some positive direction? So we thought that this could be provide us a good opportunity to investigate how robust the species barrier are. This is because we found an interesting stickleback habitat. So this area was just an urban area before the earthquake. Okay, so there should be a lot of building. But after earthquake, all buildings were gone. And interestingly, so this created a new pond. And our collaborator found sticklebacks in this pond. And we found this is a hybrid between two species. So one is Gasterosteus nipponicus, probably which was brought from the sea by tsunami, and another species, freshwater acreatus, which was probably brought by back wave of the tsunami. And my PhD student looked at the genome of this population for about 10 years after the tsunami. So this is a triangle plot of the genomic data. Some of you may be familiar with this, but others are not. But a single dot indicates a single individual fish. And if the fish comes here, it means that it is genetically almost acreatus. And if the fish comes here, it indicates that the fish is basically nipponicus. And if one hybrid should be plotted here, and if two should be plotted here, and the first generation of backcross to the acreatus comes here. And Takuya looked at the genome for over 10 years. And this is just 1 year after the tsunami. And you can see a lot of F2 hybrids and first generation of backcross. So probably this hybridization was induced by the tsunami that happened in 2011. But during the 10 years, this population became basically acreatus. Okay, so this, I'm sorry. Oh, how can I, oh, yes. So this suggests that basically they can still hybridize, but if you keep them in a same place, one genome is purged. So this is I think a very strong evidence that there is some incompatibility between these two populations, these two species. And even if you try to mix them, they become separated, like water and oil. Yeah. So anyway, so I think that you can agree that there are two species in Japan. And the important point for today's talk is that if you look at the phylogeny of labeled by blue and gray, and these are the freshwater populations. Blue indicates the native freshwater populations, and gray are probably introduced populations, because an old literature says that there are no sticklebacks in that pond, but recently we identified some populations. So probably somebody released. So anyway, to summarize so far, so there are two stickleback species in Japan. And interestingly, three-spine sticklebacks colonized freshwater environments and they diversified. But the Japanese stickleback is just marine sticklebacks, and they couldn't colonize freshwater environments. So we think that this is a great system for studying the genetic basis for freshwater adaptation. So we wondered what ecological factors that are different between marine and freshwater environments are important. And we spent several years to try to figure it out. And so I'll just skip those long years, but to make a conclusion, we found that the prey is important. So we conducted some meta-analysis, and one of the important difference between marine food items eating in marine environments and freshwater environments is the amount of DHA, which is a docosahexaenic acid, one type of polyunsaturated fatty acids. So in marine environments, sticklebacks are eating copepod or mysis, which contains a lot of DHA, which is an essential fatty acid. So this DHA is used for cell membrane. So without DHA, fish die. But they can take DHA from their diets. But in freshwater environments, stickleback eat, for example, insects or Daphnia, but they are basically deficient, lack DHA. So when the sticklebacks colonize freshwater environments, they are faced with the DHA deficiency. So we hypothesized that the Japanese stickleback and three-spine stickleback differ in the ability to produce DHA from other fatty acids, such as ALA. So ALA is abundant alpha-linolenic acid, which is abundant in both ecosystems. So we maintained these fish without DHA in their diets. And we killed them and we measured DHA content. And you can see that the three-spine stickleback could produce more DHA than Japanese stickleback, which is consistent with our hypothesis. And we also found that without DHA, this Japanese stickleback die within 2 months. But if we add DHA, they can survive. And three-spine stickleback can survive without DHA. So this suggests that Japanese stickleback requires DHA for their diets. And what could be the genetic basis for the difference in the ability to produce DHA? And we focused on one of the enzymes, which is important for introducing double bond. So DHA is a polyunsaturated fatty acid. So there are a lot of double bonds. And this FADS2 encodes desaturates, which introduce double bond. And one day I looked at the genome sequence of several populations, and I noticed that in three-spine stickleback, there's a FADS2 on chromosome 12, but the Japanese stickleback, it is absent. So for example, here is a reference genome sequence of three-spine stickleback, and here is FADS2. But when we mapped short reads from the Japanese stickleback, there's no reads, there's a gap. So my former postdoc, Asano Ishikawa, and I And our collaborators conducted a lot of experiments. I'm not showing all of the data, but to summarize, what we found is that FAS2 is located on X chromosome, only on X chromosome in the chimpanzee stickleback. So male have just one copy and females have 2 copies. But in the 3-spine stickleback, there is a copy and a paste and transposition from X to chromosome 12. So males have 3 copies and females have 4 copies. And interestingly, some of the freshwater resident populations showed tandem duplication on the X chromosome. So for example, in some populations, males have 4 copies and females have 6 copies. And finally, we conducted a transgenic experiment, and we confirmed that the introduction of extra copy of FAS2 and the chimpanzee stickleback can survive, can rescue the survival and without DHA in their diets. So this suggests that the copy number of FAS2 and the expression level, and we confirmed that the FAS2 expression is higher in the 3-spine stickleback compared to the chimpanzee stickleback. So we confirmed that the copy number and expression level of FAS2 is important for survival without DHA in their diets. And now we are conducting more refined genome assembly of several stickleback populations and species. And Yo is now doing a lot of Hi-C and Hi-Fi in collaboration with Aotearoa in our institute. And for example, if we looked at the phylogeny of FAS2, we confirmed that all of the FAS2 on chromosome 12 is making a cluster. So which indicates that probably copy and paste transposition from X to chromosome 12 happened just once in the history of stickleback. In contrast, interestingly, FAS2 copies on chromosome X are making clusters within each geographical regions. For example, Spanish populations have 5 copies, but they are making clusters. And GIF populations have 3 copies, but they are making cluster. So probably this tandem duplication on X chromosome happened independently in different regions. So now we are interested in what genomic structure enabled copies on X chromosome experience tandem duplication. And this is a dot plot. And if we looked at the dot plot, we found some interesting repetitive sequences around FAS2. And PhD student Jun Kitano found, for example, in the Spanish population, freshwater populations, there are 5 FAS2 copies on the chromosome X chromosome. And you can see some strange repetitive sequences or some inverted repeats around FAS2. So we speculate that this structure may induce tandem duplication or copy number variation, so by non-allelic homologous recombination. But of course, some experiments are necessary to prove that. We cannot exclude the possibility that this is just a byproduct of tandem duplication. And basically, this repetitive sequence is a composite transposition, transposon, I'm sorry. So this is not a complete transposon, but this is some mixture of transposon. So we are now trying to figure out what are these strange, mysterious structures. But anyway, so we found this phenomenon in stickleback, increase of copy number of FAS2 in freshwater populations. So in order to confirm the generality in fish, as I mentioned, freshwater colonization happened in multiple lineages of fish. And there are 48 species of fish whose whole genome sequences were available. So Jun, in a way, counted the number of FADS2 in these 48 species. And surprisingly, we found that this phenomenon is not specific to stickleback. For example, typical freshwater fish, such as medaka or zebrafish, have multiple copies. For example, Japanese medaka have 3 copies by tandem multiplication on 1 chromosome. And in contrast, interestingly, typical marine fish have, such as tuna, have just 1 copy. So there's a very, very clear trend. And we confirmed this is significant, even taking phylogeny into account. Okay, so what we learned from this study is that first, we may be able to predict the adaptability of organisms from genomic sequence data. For example, whether this fish can become a freshwater fish or not, based on the information of copy number of FADS2. So this may be useful for aquaculture or fisheries. And another lesson that we learn is that we can get insights into an important ecological factor, which can be a selective agent for something, for example, freshwater colonization. So before starting this experiment, before starting this project 10 years ago, I had no idea that the DHA is such an important factor in ecology. So I think this kind of ecological genetic study can help to understand what is an important selective agent acting in natural environments. So based on this study, so we wondered whether we can apply this lesson to mammals. So in mammals, so we focused on the diets. So you know that in mammals, there are herbivores, carnivores, and omnivores. And I agree that it is very difficult to make a clear line, but it is difficult to distinguish among them. But there are some spectrum of diets. But anyway, so there are mammals using different diets. And a previous study suggests that transition between diets happened multiple times in multiple lineages. But this happened only in a limited phylogenetic lineage. So we think that this is a very similar situation that happened in freshwater colonization fish. So we wondered what kind of gene, copy number of what kind of genes are associated with diet shift in mammals. So we one intern joined my laboratory, and we established collaboration with Iwasaki Laboratory in University of Tokyo, and his student Shun Yamanouchi helped a lot. And what we did is that first extract, compile diet data of mammals, and Iwasaki Laboratory is very good at making ortholog groups. So there are about 63 mammalian species whose whole genome sequences are available, and they are good annotation, protein annotation. So they made an ortholog groups. So in some cases they are ortholog, or in some cases they are parallel. But so they cluster genes whose protein products are similar. Okay. And we made some arbitrary criteria. And of course, this is just arbitrary. So by changing criteria, the absolute number of genes that are detected become different, change. But anyway, so we conducted some first screening based on some ratio, making a threshold of ratio, and which is associated with diet. And finally, we conducted some phylogenetic correction. So usually, phylogenetically closely related species have similar ecology and similar copy numbers. So we need to correct phylogeny. So in conclusion, so we found several genes whose copy numbers are associated with diets. And for example, here are some examples. Herbivores increased some genes encoding enzymes involved in detoxification. So for example, plants produce toxins because they are eaten by some animals and they try to avoid predators. So there is an arms race between plants and herbivores. And so some enzyme introduce some glutathione or gluconic acid or something like that to the toxin, and which makes them soluble, and which is excreted into urine or bile acids and get secreted out of the body. So it makes sense that the herbivores increased copy numbers of these genes. And interestingly, in carnivores, we found that the genes involved in iron-induced oxidative stress increased. For example, ferritin or some other cytochrome oxidase. And this is just speculation, but we think that red meat contains a lot of iron, and that can be an oxidative stress. So for becoming carnivores, there should be some adaptation to that iron overload. For example, here is an interesting story. So in Africa, some people are making beer in iron pot. So they take too much iron, and then it causes several symptoms, and it is called African iron overload. So if you Google on the internet, you can find this information on Wikipedia or something like that. Yeah. So anyway, so taking too much iron is not good. And omnivores, we found an increase in amylase gene. And this is not new, previously reported, but it is good that we could confirm the previous study. So anyway, so in the case of mammals, we cannot identify a single gene. So like a FAST2 in freshwater colonization in fish. But we wondered whether we can make some function, linear function, which can distinguish carnivores and herbivores based on copy numbers of several genes. And by stepwise selection, we could reduce the number of genes that are important to 14 genes. So just by counting the copy number of 14 genes, we could distinguish carnivores and herbivores. And interestingly, based on this score, omnivores show broad distribution. So we think that this is helpful. And interestingly, some omnivore is has a very high score, and which is green monkey. And in our study, this was classified as omnivores, but they are eating a lot of plants. And some omnivores have very small scores, and these are red fox or European shrew, and they are eating a lot of animals. So we think that this score is saying something real. And by the way, human is just between carnivores and herbivores. So maybe, perhaps humans should eat a good mixture of vegetables and fish or meat. But anyway, so based on these results, I would say that we may be able to predict the adaptability or ecology of organisms from genomic sequence data. Yeah. And we can also get important insights into an important ecological selective agents. For example, perhaps, maybe iron in the diet is important constraint for making animals carnivores. So before starting this project, I had no idea that the iron is such an important element. Of course, we know that sometimes humans become iron deficiency. So we think that taking a lot of iron would be good. But as I showed in African iron overload taking too much iron is not good. Okay, so now I'll move to the morphological evolution. So again, I'll go back to the stickleback story. And when marine sticklebacks colonize freshwater environments, sometimes they reduce the armor plates. So this is a bony structures, and now alizarin red stains calcium in these two specimens. And you can see that the marine sticklebacks have complete sets of armor plates, but freshwater stickleback reduced the armor plates. Maybe this is adaptive. At first, they can, in freshwater environments, calcium are deficient. So they can save calcium and they can allocate calcium to more essential parts. And it can increase buoyancy. So you know that in the sea, you can float, but in freshwater, it is difficult to swim. So increase of buoyancy is important. And in some freshwater environments, there are a lot of vegetable, not vegetable, there are a lot of plants, so they can escape into these regions. So rather than having armor, it would be suitable for increasing the escape response, swimming ability. So anyway, so this happened in Europe, North America, and also in Japan. Okay. So this is a good example of convergent evolution or parallel evolution. So we are interested in the genetic basis for this. And previously, David Kingsley's group in Stanford University has identified that either ectogysplasmin A is responsible for plate reduction in North American and European populations. So marine sticklebacks have one type of allele, and freshwater stickleback have another type of allele. And they found that there are two major alleles. And as I mentioned, marine sticklebacks have basically one type of allele, and they are sometimes heterozygote. But in freshwater environments, another alternative allele become predominant. And this polymorphism is maintained by selection-migration balance. But interestingly, so Japanese populations have complete either allele, although they are low-plated. So we are interested in what are the genetic basis for the plate reduction in these Japanese low-plated fish. So Yamasaki and we collaborated with Tomoyoki Kokita, and we conducted several genetic analysis. For example, we crossed Japanese marine stickleback, completely plated stickleback, and GIF low-plated stickleback, and we conducted QTL mapping, and we found two peaks. And one corresponds to either, which explains only 26% of phenotypic variance. So in the European and North American populations, that canonical either allele explains about 75% of phenotypic variance. And we also found another peak, and which explains 46% of phenotypic variance. And interestingly, this was either receptor. So either is a ligand, and which binds to this receptor. And I'm sorry, I'm not familiar with molecular mechanisms or molecular development. But anyway, so this induces something, and it causes some development of armor. Yeah. And we confirmed that this either receptor is expressed where the plates are formed. And Yo made a knockout sticklebacks, and we confirmed that the knockout either our knockout sticklebacks lack the plates. So this suggests that this is a responsible causative genes. And This is another low-plated population in Japan. So in 1987, there are only low-plated fish. But fortunately or unfortunately, somebody released a completely plated stickleback in this region. So they hybridized. And now they show great variation in phenotype. So by using this, we can do GWAS. And we found, again, two peaks, and one was EDA, and another was EDA receptor. Okay, so based on this study, we found that the EDA-R signaling pathway is very, very important for plate reduction in the three-spine stickleback. Next, we looked at another genus, which is Pangitis. So in this closely related genus, we can again found some polymorphism of plates. Some are completely plated and some lack plates in this region. And we again found some polymorphic population. So by using this, we could conduct GWAS, and the peak was located on EDA receptor. And Yo found that some region is lacking in the partial mode because of the insertion of retrotransposon. So we hypothesized that this 600 base pair may be important and enhancer for plate development. So he deleted this region by CRISPR, and we confirmed that this deletion can cause a plate reduction. So in this Pangitis, we could identify a causative mutation, probably transposon insertion breaks the enhancer, and which is responsible. So anyway, so this suggests that in multiple independent lineages, very similar genes in the same signaling pathway is repeatedly used. So there are some predictability in the genetic causes of convergent evolution. But anyway, they use different alleles. So the final question that we are trying to ask is, yeah, they use the same genes, but are there any difference in the function of alleles? So as I mentioned, in the North America and Europe, there is a lot of gene flow between marine population and freshwater population. So this polymorphism is maintained by selection migration balance. In contrast, in Japan, basically freshwater populations that I mentioned, Gifu or Shiga, are isolated from marine environments. So there is no gene flow. And there is some previous study which indicates that gene flow with divergent selection can promote the linkage of adaptive alleles. For example, if multiple adaptive alleles are making cluster, which behave like a super gene, and then the net selection at the locus becomes very strong. Okay, so basically, gene flow has a homogenizing effect. So in the presence of gene flow, allele frequency differences between two divergent environments should be homogenized. But if there is a very, very strong selection, that can maintain difference in alleles between two environments. So if they make cluster, they are linked, they behave as a super gene, and the net selection becomes stronger, and then they can be maintained as different alleles in that population. So this was a theoretical prediction. And actually, David Kingsley's group confirmed that this canonical EDA allele is linked with QTL for many other morphological traits. So this is something like a super gene. They call super gene, but of course, I know that the definition of super gene is difficult, so I'm not sure whether we can call this super gene. But anyway, so this region contains QTL for many traits. So this is consistent with the prediction, theoretical prediction. But as I mentioned, in the Japanese populations, for example, Gifu and the marine populations have very, very low gene flow rate. And Nasu and the marine populations, basically there's no gene flow today. And we conducted some simulations. Yo Yamase conducted some simulations to confirm that. So we hypothesized that Japanese EDA allele is not linked with many other traits. To confirm that, we measured all of the traits that were reported in a previous study. And we found that basically none of these traits were mapped on EDA or EDA receptor. So which suggests that although the same genes, EDA, is used in the Japanese populations, North American EDA allele and Japanese EDA allele have different functions. Okay, so this is a basically last slide to summarize what I reported today. So of course, I do not have a clear answer, but I think that genetic basis for convergent evolution is predictable to some extent. And because of that, genome sequence data is useful for the inference of adaptability of certain organisms, whether this fish can become a freshwater fish or whether we can maintain this fish in freshwater or not, something like that. But of course, historical contingency is also important. So Japanese populations and other populations use different alleles, and they have some different functions. But as I mentioned, we can predict why North American populations has multiple functions, but Japanese EDA allele is not linked with other traits based on the information of the extent of gene flow. So yeah, we hope that further investigation on the genetic basis of convergent evolution would help to understand the genetic basis for convergent evolution, determinism and the contingency of genetic basis of convergent evolution. Okay, so finally, so I'd like to thank all lab members who produced all of the data that I'm talking today. And I also would like to thank the funding source supported my research. And I'd like to thank all audience for listening to my talk. Thank you very much.
Just want to take the opportunity to thank you again for your time. That was a fantastic presentation and really interesting research. We look forward to following how it transpires in the future. And yeah, everyone's clapping and saying a big thank you. So with that, I think we'll end the session.